Hello, and welcome to this RPTH Research Recap, where we're going to dive in a little bit deeper into some of the science that's being published in RPTH. So my name is Erica Sparkenbaugh, and I'm an assistant professor at the University of North Carolina at Chapel Hill, and I'm a member of the Blood Research Center here. My lab investigates endothelial dysfunction and inflammation in red blood cell disorders. And I'm also one of the social media associate editors at RPTH. So I get to see a lot of the science that comes through. And I'm really pleased to introduce Dr. Melissa Chan today. Dr. Chan is a research fellow in the lab of Dr. Andrew Johnson at the National Heart, Lung, and Blood Institute's Framingham Heart Study in Massachusetts in the United States. Dr. Chan received her PhD in cardiovascular pharmacology from Queen Mary University of London in the UK and conducted postdoctoral training at Imperial College London and at McMaster University in Canada. She then returned to London to specialize in the role of antiplatelet therapies. Her research interests lie in optimizing platelet reactivity testing, particularly in large and understudied populations, by developing novel and improving existing assays. Dr. Chan recently published a paper in RPTH titled Factors Which Modulate Platelet Reactivity as Measured by 5 Assay Platforms in 3,429 individuals. So in this study, Dr. Chan utilized 5 different approaches to evaluate platelet function in a large cohort from the Framingham Heart Study. And they evaluated different platelet agonists. They compared a lot of different factors that might contribute to outcome variability in these assays. So it was a really comprehensive study, and I'm really looking forward to hearing more about it. So for those joining us live, please leave your comments, which will be answered during the discussion. Dr. Chan, welcome, and we look forward to hearing your presentation.
Thank you. Let me see. Okay, so thank you to the RPTH for allowing me to share some of our work that we've been doing up in Framingham. So I don't need to tell anyone that platelets play a major role in hemostasis and thrombosis. So they're small but mighty. The platelet function is actually, so the increased platelet function is actually associated with increased cardiovascular disease. And the assessment of platelet function is actually critical in diagnosing bleeding disorders. However, it's quite tricky. So the gold standard is light transmission aggregometry, so LTA, which was actually developed in the 1960s by Gustav Born. And it's great at looking at platelet function. However, it does require experienced staff. It's time-consuming, labor-intensive, and a relatively large volume of blood is required to do it. And so actually, like large-scale population studies are quite rare. So there have been some genome-wide association studies looking at genes and light transmission aggregometry outputs, but only in kind of a small number of agonists. So here's ADP, epinephrine, arachidonic acid, and collagen. So we've done this in the Framingham Heart Study, looking at 2,604 European ancestry people. And it's also been studies in the GeneStar population, which is African American, and there's been replicated in other studies. Looking at kind of an expanded platelet function, so looking more deeply at the phenotype or a broader brush approach, has only been really done by me when I was in Tim Warner's group in London. And that was in the Bruneck study in 338 elderly individuals. So what we wanted to do was to utilize the Framingham Heart Study cohorts, which NHLBI funds. So for those who don't know, the Framingham Heart Study started in 1948 with the original cohort being recruited. Then we had their children, so they're called Gen 2, and also now we're on the 3rd generation, so the grandchildren of the original cohorts. And every couple of years we bring them in and we do physical examinations, blood samples, and also collect some other data as well. So the aims of this paper were to look at platelet reactivity using a couple of different platelet assays in the Framingham Heart Study cohort, and additionally to identify any technical aspects during the data collection which actually moderate these values coming out of these tests. So these are the assays that we used. So first I'll talk about the gold standard, so light transmission aggregometry, which is the fourth panel along here. So this measures a change in light transmission after the addition of an agonist. So in this left picture, you've got your cloudy platelet-rich plasma. So what we do is we take whole blood and then we centrifuge, and we can take off the top layer, which is the platelets in plasma. And this is a cloudy mixture. So what we do is put that into a cuvette with a little stir bar and shine a light through. And at the back here, you have a photosensor. So because this is a cloudy mixture, you have not very much light going through. But as soon as you add an agonist, all these aggregates form and take them away from the cloudy mixture. And so you get more light transmission across. So more light transmission equals more aggregation. So using the optimal assay, we kind of flip this on its side. So instead of adding the agonist to the platelet-rich plasma, we put the agonist and freeze dry it onto a 96-well plate. And then we add platelet-rich plasma to it. And then we can read absorbance. And instead of having stir bars in each individual well, we physically shake the whole plate on a plate shaker. So the advantage of this is that we can make concentration response curves to multiple agonists at the same time. In addition, we also looked at multi-plate impedance aggregometry developed by Roche in whole blood. So this just measures resistance across two wires. So the more aggregation you get, the more build-up between these two wires and more resistance. We also use this TTAS system, which flows blood across a collagen-coated microchip. So we can see the build-up of aggregates in these capillaries, and when there's an increased pressure, when it's blocked, then that's a higher thrombus formation. Finally, we use flow cytometry in whole blood and platelet-rich plasma, and we stimulated with ADP and looked at the expression of activation markers, p-selectin and PEC1 binding. So looking at the cohort, as I said, we used generation 3, so the children of the original cohort. We also, in this study, we also had Omni cohort, which is a multi-ancestry group, and then the new offspring spouse, so NOS. And we classified aspirin use as the final aggregation in LTA to arachidonic acid, giving less than 40%. So here we've got a spread of people, of males not on aspirin and on aspirin, and females not and on. So the first thing we wanted to do was to look at the correlation between these assays. So looking at LTA versus LTA. So here we've got LTA, optimal, multiplate, TTAS, and flow. So all of these assays, and then when you look closer, you have the different outputs. So aggregation to different agonists. So these are mostly for activation, so aggregation. So with a dark red here, you have a positive correlation. So this here is a complete, like perfect correlation. And then with blue, you have a complete negative correlation. So we're all going to be on this the scale at some point. So as you can see, there's a reasonable amount of reds and blues that are going on here. And what's important to note is that this correlation matrix was actually using every one of the participants who had platelet data. And we know that aspirin really strongly affects platelet function. So what we did was to do this again using the Pearson's correlation, but removed those 736 aspirin users. So with this, you can see, you kind of get a clearer picture. So we still have that high intra-assay correlation. So LTA versus LTA, as you can see, like going down the diagonal. And we also noticed that there was a low inter-assay correlation. So we're looking at multiplate arachidonic acid versus LTA arachidonic acid and optimal arachidonic acid. There's very minimal correlation, if any. So what we can say here is one assay might not be a surrogate for another. So the next big thing that we wanted to do was look at the pre-analytical medication and technical factors. So these are things that haven't really been studied in this kind of detail before, but we love data, so we're going to do it all. So we looked at age and sex, platelet-rich plasma volume per citrate tube. We looked at medication use. Also, those technical factors, so deviations from a blood draw. Did they need 2 sticks? Or were they feeling faint? We also looked at the time of year, as well as the time of the draw and the time from draw to assay. And then agonist batch, we tried to limit our batches as much as possible to kind of 1 to 4 batches for the entire exam, which was 4 or 5 years long. We also noted the technicians, so the phlebotomists and the assay technicians. And we analyzed using a linear mixed effect model. So with this, we've got our different assays across the top, and then our kind of different technical factors down the bottom. So just to explain this this plot to you, the bigger the circle, the smaller the p-value, so the more significant. And for those with directional effects, orange is a negative effect, and a blue is a positive effect. So if you look at age versus LTA, where you have these big blue circles, that suggests that with these LTA traits, you actually have an increased aggregation as you increase age. Conversely, when you have aspirin use, you have a decrease in aggregation. So, you know, p-values are well and good, but actually how much does it affect the number itself? So that's where we looked at the variance. The proportion of variance is how much of the kind of final number is it affecting. So here you've got the bigger circles is more than 10% of variation, which is enormous. And then, you know, we're going all the way up to the highest red, which is around 50%. And you can see that aspirin use is really contributing about 50% of the of these outputs. So in summary, aspirin strongly affects platelet function, and age and sex really contribute to these measures. Mostly they're enhanced, but sometimes they're variable. So it's important to note what's going on there. We looked at technical factors, and the ones that are really affecting platelet function assays, or at least the numbers that come out at the end, are agonist batch, the technician for the assay, and the day and year. So what is really like our take-home point is that platelet assays aren't interchangeable, and really one assay can't be exchanged for another. And finally, we need to consider technical factors when interpreting the assay data, which is something that we haven't really thought about before. So I just want to thank our collaborators, the Framing Heart Study participants, and you for your attention. Thank you.
Thank you, Dr. Chan. That was a really great presentation. It really helped clarify the data for me. Before I get started on my own questions, I just want to remind the audience that please, if you have questions or comments, please put them in the chat and I can address those. But while those are coming in, I have a few questions about the study that came to mind when I was reading the paper. So given the findings, how do you think future studies could be designed to account for the variability in the platelet reactivity measures?
Okay, so
assays.
Yeah. So this always depends on what your study design is. So there's no like perfect answer. When you're looking at larger population studies like the Framingham Heart Study, there are things that you can't control. You can't control what time of day you're going to draw the blood. You can't control who's doing it usually. But you can account for those things. And you definitely can account for things like age, sex, and aspirin use. Those can be very clear readouts that you have access to. And it's really just looking at how you're analyzing things and how things are balanced, really. So for smaller studies, so if you're looking at different cohorts and you're giving treatments, things that are already done, age, sex match. But yeah, just noting other things that are going on. So, have they been smoking? But that also will affect platelet reactivity. And then for the smaller mechanistic studies where you've got, N of 20 healthy volunteers, those things you can start accounting for. You can account for batch. You can do, age, sex match. You can have the same person do the same assay.
Yeah, that was a question I had. Like, do you have, for the study, do you have the same person running? Like, is there one person dedicated to running the LTA versus the other, like the flow cytometry?
Yeah, so for these, the study went on for about 4 or 5 years.
Yeah.
And the current one is also going on. So we have like a rotation of people who can usually do all of the tests.
Good.
And we try to split it up as much as possible. There's things we can't account for, the learning curve for LTA, for example, your reproducibility.
Yeah.
But yeah, we try to split it up as much as possible because I've done a lot of LTA and one person's not going to want to do it every day of their life.
I know. Well, speaking of LTA, we actually have a good comment from the audience. Do you think different LTA platforms have better comparative performance?
So this is the thing about LTA. We call it LTA, but every single clinic, every single lab will have slightly different setup. So even if you're comparing, the chronolog to biodata, right, the mechanism is the same. You're measuring light. It's just like shining a torch through and then seeing how much is on the other side. What is more important there in comparing them is looking at your agonists and your agonist concentrations. And that's the other thing that we need to consider is that isn't standardized across the world. So I think what is really important there is having that healthy, person that's free of NSAIDs for 2 weeks and having that direct control.
Yeah.
I think that's more important definitely in diagnosing and in tailoring antiplatelet therapies.
Yeah. So I had a question that kind of dovetails from that. I'll ask that and then maybe I'll go back to the chat. So my second question kind of addresses that. How might these findings impact clinical practice? So for diagnosing bleeding disorders or evaluating like a new antiplatelet drug efficacy?
Yeah, so I think that, along with what I just said, we need to, think about looking at age and sex match controls, especially for LTA. So I've shown that in females, for example, you tend to have a higher percent aggregation than males. So, if you're looking at efficacy of drug, your female might just have a higher response in the first place. But if you're comparing to a male who has a lower response, it looks like there's been no effect of the drug.
I see.
So, that's important. It's important to, to think about how the interpretation will lead to, will guide treatment. So just looking at all the factors that might be involved as well.
Yeah. And I think that actually addressed one of the questions in the comments from Hira Niazi. What about comorbidities such as diabetic patients and platelet reactivity? I think you really addressed that by having the right controls.
Yeah, that's something that we're working on right now as well. So diabetes is also, kind of a multifactorial thing as well. So, we'll class someone as diabetic, but actually, are they on anti-diabetic medication? Are they on insulin? Like, there's lots of different little things that can be involved, and that's all to do with your analysis model. But yeah, we have seen actually that people with diabetes have a slightly different platelet reactivity, and it's looking at those different pathways. So the great thing about having these 5 different platforms and different agonists is that we can see that people with diabetes definitely have different pathways that are either upregulated or downregulated. So we can maybe try and target those ones using medications.
Okay. There's another question in the chat. Congratulations on this magnificent work. In the future, do you plan to test the inflammatory response of blood platelets, in particular the release of classic soluble factors such as soluble P-selectin, CD40 ligand, and RANTES?
Yeah, we'd love to have your input on things like that. In our current analysis, so we're doing the exam 4 that's coming through now. We are doing a kind of expanded panel of flow cytometry. So we're looking more deeply at leukocyte and platelet leukocyte interactions rather than looking at the inflammatory response per se. The issue with collecting this data is that we have to do it on the day, and we'll get 6-7 people, and it's a time crunch, especially if you want to do everything within kind of the ISTH guidelines of 4 hours. So it is all hands on deck, and we would absolutely love to do kind of those more mechanistic studies, which I think we're going to try and do with our lab down in Bethesda.
That's amazing. That's really helps help me visualize what these lab days must be like, trying to get all the samples processed. So this was a really fantastic chat today. Thank you for all the questions. I have one final question. So, I am not a platelet biologist, and I don't really know a lot about platelet function tests. So could you offer some advice for like a basic scientist who doesn't have a lot of background in platelet biology, but wants to investigate some platelet function assays in their study population? So maybe of the 5 assays, could you recommend maybe 2 assays to start with that a lab could undertake to get an idea?
Yeah, so I mean, the first thing is definitely look at the ISTH guidelines. Those papers, those SSCs have, so many hours of work put into them, and they do have like some great recommendations. For the assays that you suggest, or I would suggest using, I'm always gonna, promote the optimal assay. I think it's for a broad brush, like overview of what's going on in the platelets, it's great just because it takes 5 minutes. You don't really need to be trained very highly, and you kind of use most of the instruments that you already have in the lab. So an absorbance reader and
that's key, I think.
Yeah, so try to make it as cheap as possible, but like showing as much as possible. It can also be tailored, so we can use different we can put different agonists on. But it's like really an off-the-shelf kind of assay. So you just need your platelet-rich plasma and your platelet-poor plasma, and you can kind of do it.
Oh, great.
Also, like, you don't need very much blood compared to LTA. I will say that with the optimal assay, like in order to do that deeper dive, then yeah, LTA is the one that you have to use to prove to everyone, as it's the gold standard, that what you're seeing is what you're seeing. Yeah, I love the LTA too.
Thank you. Thank you so much. Well, thank you, Dr. Chan. This was really wonderful. And thank you for presenting your work and giving us more insight into this paper. I'm so impressed with the size of the data set and just the amount of work that went into getting this paper out, and just all of the findings that you were able to contribute to our field. Thank you so much. I think the viewers can all appreciate the amount of work that went into this study too. Thank you.
It's a village.
All right, we hope the viewers from today will read the full details of your paper. They're on the site rpthjournal.org. Thank you for those of us who joined live, and don't forget you can follow RPTH on Facebook, LinkedIn, Twitter, X, and on YouTube. The recording from today will be found on YouTube. So thank you all for watching, and keep an eye out for the next RPTH Research Recap. Thank you.
Thank you.